Welcome to Biology with Deepika. In this video, we will learn about the part 3 of sexual reproduction in angiosperms, and we see in details the structure of ovule and the process of megasporogenesis and the formation of megaspore. So here in this picture, we see how the ovary is here, and this is the ovule is placed inside the ovary. These are the egg cell, and this is the polar nuclei, and these are the antipodal cells, and how these cells are formed. The part 1 and part 2 of this video have microsporogenesis and the process of microgametogenesis and pollination. The link is provided above and in the description box. And in this video, we will learn about the process of megasporogenesis, that is the formation of megaspore from achisporial initial, and the megagametogenesis, in which the female gametophyte is formed from megaspore. So let's see in details. First, we will see how the structure of ovule look like and what is the ovule. The process of megasporogenesis, that is the formation of megaspore from megaspore mother cells, take place in ovule. This is the ovule, and the process of megasporogenesis take place here. And this is known as integumented megasporangium because it has two integuments, the outer integument and the inner integument. And the ovules are situated inside the ovary. Here we see how the ovule is situated inside the ovary. This is the ovary, this is the ovule. The ovule is situated inside the ovary, and it is connected through placenta. And this is the special tissue. The placenta is a special tissue. And when we go to in details the structure of ovule, first we will see the funicle. This is the funicle. The funicle is the point of attachment of the ovule. One side it is attached to the body, and the another side it is attached to the placenta. When it is moved upward, this is formed the chalaza length. This is the chalaza, and this ridge is formed is known as the raphe. This end is the downward end is known as the micropyle. This is the micropyle, and this inner tissue is known as the nucellus. This is the egg apparatus. This cell, these three cells together is called egg apparatus, and these two nuclei are known as polar nuclei, and these three are known as antipodal cells. So this is the embryo sac. So this is all about the structure of ovule. And conditions seen in ovule in relation to integuments are unitegmic, in which one integument is formed. It is found in sympatalous or gamopetalous dicots. These are the examples. And in bitegmic type, two integuments are found, and it is found in polypetalous dicots and monocots. And a reel is found in lychee, nutmeg, etc. This is the important example. A reel is found in lychee, and carbuncle is found in castor. This is also important, and Euphorbiaceae family. And ategment means no integument is found. This is found in Loranthus viscum and santalum. This is all about the ovule, its structure, and how the different type of integuments are found. The next we will see how the different type of ovules look like, its structure, and with examples. The first one is the atropous or orthotropous type. In this type, micropyle, funicle, and chalaza are situated on one vertical axis with micropyle directed upward. Here we see everything is present at one axis directed upward. This is known as orthotropous or atropous, and it is found in polygonum and piper. The second one is known as anatropous. It is known as the inverted ovule because the body of the ovule is so bent that micropyle is now directed downward and lies close to the hilum. Here it is directed downward, micropyle, and it is close to the hilum. This is the hilum, and this is known as inverted type. This is the anatropous type, and 70% of angiosperms mostly have this type, anatropous type. The third one is the compylotropous. In this type, it is placed, the ovule is placed transversely on the funicle. Here we can see how the ovule is placed transversely on the funicle. This is the funicle. The ovule is placed transversely so that the body is bent such a way that micropyle gets directed downwards and is not in straight line with the chalaja. It is found in gram, beans, member of crucifera like mustard, etc. And the fourth one is hemianatropous. In this type, the body of the ovule is situated at right angles to the funicle. Here we see how the body of the ovule is placed right angle to the funicle. This is hemianatropous type. For example, it is found in ranunculus. The fifth one is known as the amphitropous type. In this type, the ovule is curved in such a way that embryo sac also becomes horseshoe shaped. Here, amphitropous, embryo sac becomes horseshoe shaped. The micropyle is directed downward and doesn't lie on straight line with chalaja. For example, we can see these in poppy, members of Alismaceae, Butomaceae, etc. And the last one is the sarsinatropous type. In this type, the ovule is initially orthotropous but later becomes anatropous. Here we see sarsinatropous type. The growth continues till the ovule once again becomes orthotropous. This is found in Opuntia. And this is how the different type of ovules look like. And the most common question we can found in exam are the atropous or orthotropous type. This is directed upward and found in Polygonum and Piper. And anatropous type is inverted and is found in angiosperms. And we can also get, which is important in exam, is horseshoe shaped ovule is amphitropous. So this is all about the type of ovule with proper diagram. Here we can see. Next we will move on to the process of megasporogenesis. In this process, megaspore is formed from archisporial initial. In this process, it begins with the differentiation of archisporial initial in the nucellar hypodermis. Here we see. In the nucellar hypodermis, it begins and the megaspore mother cell is formed. This is the parietal tissue. Archisporial initial either directly acts as megaspore mother cell or divides periclinally into an outer parietal cell and inner sporogenous cell. According to this, it is distinguished into two types. There are two conditions found. First one is tenunucellate ovule found in gamopetalous dicots with unitegmic ovules, in which directly the archisporial shall act as the megaspore mother cell here. But in crassinucellate type, in the second one is known as the crassinucellate type. In this type, the archisporial initial either act directly as megaspore mother cell or divides periclinally into an outer parietal cell and inner sporogenous cell. Here, this is the crassinucellate type. Here, the primary parietal cell, archisporial initial is divided periclinally into primary parietal cell and primary sporogenous cell. This is found in polypetalous dicots with bitegmic ovules. Then the megaspore mother cell divides meiotically. This megaspore mother cell, to form 4 megaspores, which is haploid. Out of these, 3 degenerates, and the lowermost megaspore present at the chalagial end act as the functional megaspore. And the megaspore is the first cell of female gametophyte and it is haploid because it is formed due to meiosis. Then we will see the process of megagametogenesis and the formation of female gametophyte. The process of development of female gametophyte or embryo sac from megaspore is called megagametogenesis. The embryo sac can be monosporic, bisporic and tetrasporic depending upon the number of megaspore taking part in the process of development of embryo sac. Here we see only one functional megaspore is formed and this take part in the formation of female gametophyte. So it is known as monosporic type, and If two spores participate in the formation of female gametophyte, it is known as bisporic. If four participate, it is known as tetrasporic. The most common type is monosporic 8-nucleate embryo sac known as polygonum type and occurs in 70% of angiosperms. This usually comes in the exam in objective type. And the organization of monosporic 8-nucleate polygonum type is as follows, and we will see how it looks like. First is the egg apparatus. Here we see the egg apparatus. These three cells are together called egg apparatus. The middle one is known as the egg cell, and the two side cells present in the side is known as the synergids. A filiform synergids show a filiform apparatus attached to their upper wall, which helps to attract and guide the pollen tube. Here we see the filiform apparatus is present in the synergids, and this helps to guide the pollen tube. A beak is also present here. Here we see in egg cell the nucleus is present downward and the vacuole is present upward, but in the synergids the vacuole is present downward and the egg cell and the nucleus is present towards the upper side. Then the second one is known as the polar nuclei. This is one cell. This is binucleate. Both the polar nuclei fuse before fertilization and form a single diploid nucleus called secondary nucleus or definitive nucleus. The polar nuclei fuse to form secondary nucleus. This might come in the exam. What results in the secondary nucleus? What fuse? So polar nuclei, it has two cells. They fuse to one cell, and this is known as the secondary nucleus, and it is found in the center of the embryo sac. And the third one is known as the antipodals, the three-celled stage. Here these are the antipodals, three cells, and found in the chelar gland. In this way it has totally 7 celled, 1, 2, 3 cells plus 1, 4, and this 3 antipodal cells, 7 cells in total. So a mature monosporic polygonum type of embryo sac at the time of fertilization is 7 celled, 3 plus 1 plus 3, and 7 nucleate type. This is all about the process of the development of female gametophyte or embryo sac from the megaspore with diagrams. Here is the diagram, and here is. And what is then apospory? The development of gametophyte from sporophyte without involving meiosis is known as apospory. In angiosperms, apospory was first reported by Rosenberg in Heresium. Since egg cells in aposporic embryo sacs is diploid, it develops directly into diploid embryo without fertilization. This is known as parthenogenesis. So this is all about the structure of ovule, megasporogenesis and megagametogenesis. Thank you for watching my video. Do like, share and subscribe if you like my video. Thank you so much.